Well, thank you for inviting me to give this talk, and it will be a talk about science and application of neutron reflectometry. But since we are all working in this field, in the neutron reflectometry field, I hope I will be able to share with you a little bit more details that I normally can when I have to give this kind of talk. And in general, for those of you who don't know me, I work at ESS in connection to the STI instrument. That is one of the in-kind contribution from PSI. But in this talk, as I said, I will not really talk about instrumentation, but more about scientific application of neutron reflectometry to biological membranes. And this is kind of now became my standard slide when I have to start to talk about biological membranes. And this is a simplified version of what a biological membrane looks like, where I try to highlight the fact that it mainly contains two components, proteins and lipids. But despite this, there is quite a large variety of different proteins and hundreds of different lipid species that are all mixed together and forming this very complex structure. This compositional complexity makes it quite difficult to study biological membranes directly as they are in a cell. And there are also quite a lot of complication eventually related to extracting these membranes directly from cells. So what happens is that over the years, simpler model systems have been developed, and supported lipid bilayers are the one that I'm going to talk about, as they're also like perfect candidate for characterization with surface sensitive techniques, and in particular neutron reflectometry. So normally supported lipid bilayer are formed using a relatively simple composition, perhaps 1 to 3 phospholipid species is what you normally find in most publication. And there is quite some reliable protocols that you can use to actually deposit them on solid substrate. One of these protocols basically starts with lipids dissolved in solution in the form of vesicles, and then exploiting the fact that this vesicle will spontaneously fuse on the support surface and form a bilayer, as you see represented here. What I am personally interested in general is to try to make these kind of systems a little bit more complex and therefore closer to biological membrane or to the corresponding membrane that those systems should mimic. And in particular, I have been working with either systems where we used a natural lipid extract to form this supported lipid bilayer. And this means the lipids that were directly extracted from cell. These extracts normally contain quite a large variety of different lipid species as you would normally find in biological membranes. And then also the possibility of not just having lipids deposited on your substrate, but also have membrane proteins incorporated in these bilayers. And a part of the goal of my talk here is to show you that it's kind of not that easy to produce this system. Sample preparation is actually quite a big part of planning to do neutron reflectometry experiment on this system. And I would like then to show you a method that we have recently developed for producing this more complex supported bilayer. So in this presentation, as I mentioned, I will start by showing you how supported lipid bilayer containing complex lipid composition and even incorporating membrane proteins could be produced. And then I show you some example of different lipid composition that we have tested. And then in the end, talk about a more recent case that we have investigated, where we use these supported lipid bilayers to investigate the effect of spike proteins that are found on the SARS-CoV-2 virus. So kind of like a hot topic, not in these days. So to form the supported lipid bilayer, instead of simply injecting lipids on the substrate and let them self-assemble, we developed a different approach that is based on the use of peptide disc. These peptide discs are discoidal particles that contain an inner core of lipids organized as a bilayer and are stabilized in solution by this red belt that is made of those self-assembled peptide molecules. These kind of particles were initially developed for studying membrane protein in solution, but it was soon found out that they are very dynamic. This peptide comes and goes from the lipids, and they tend to rearrange and fuse quite fast. So in the end, they did not have a lot of success for studying in solution. What we thought of doing is to actually use these peptides to bring lipids to a solid surface and exploit the fact that they can rearrange to then fuse them together and form a continuous supported bilayer. And this is possible because this peptide works a little bit like a detergent. So the peptide that is bound to the lipid is actually also in equilibrium with the free peptide in solution. And by rinsing with buffer, it is possible to remove the free peptide from solution, therefore favoring its release from the lipids, which eventually leads to the formation of a continuous bilayer. What I found really interesting about this method is that it has only constrained the fact that the support surface has to be hydrophilic, which then means that the same peptide disc solution can be used to form supported lipid bilayer on various kinds of substrates, and then is therefore compatible with different kinds of surface sensitive techniques. And here you can see a list of those that we have directly tested, but of course there could also be others. So this idea of using the peptide disc is also in connection of producing supported bilayer with membrane proteins incorporated. And this is quite interesting because if your membrane protein is asymmetric, so it, for example, has one large extramembrane domain and the rest of the protein is all buried inside the membrane, in your final supported bilayer, you would like to have that all the membrane proteins actually share the same orientation. So they all expose this extramembrane domain towards the bulk solvent. This is for two reasons. Of course, having a unique orientation will make data analysis much more easier and reliable, but it's also the way that nature works. So in a membrane, all membrane protein will have the same orientation. So this will also in the end make the system much more biologically relevant. So the protocol for forming this membrane is the same as I described before for just lipids. And now the fact is that these peptide discs are so small that when they approach the support surface, they have a certain freedom of deciding with which orientation they ended up depositing on the substrate. And it turns out that the orientation where the lipid head groups are closer to the substrate surface is actually the most favorable one, so that you end up having a unique orientation of your membrane proteins inside the bilayer. So we have actually tested this method with different membrane proteins that come from different kind of organisms, but they also have different structure, and most importantly, different sizes. So when we first tested the method, we decided to go with something very large and rigid. So we started with this bacterial magnesium transporter that is called CoRA, that has also a very well-known structure. And it was also a very good test protein because it's relatively easy to produce. So we had quite a good sample availability for doing several tests. And then we moved to something that was smaller and also more biologically interesting in the sense that there were less information available. That is the case of tissue factor, a mammalian membrane protein. And then most recently, we have tested this other mammalian protein that is called FAMP2. It is located in the synaptic vesicles, basically in our brain. So it participates to the process of neuronal communication. And as you can see, this one is now a very small membrane protein with a relatively small extramembrane domain. And in all these three cases, we were able to prove that the peptide disc method could, first of all, form supported bilayer of good quality, so with good surface coverage and retaining the orientation of the membrane protein in the bilayer. So I promised before that I will not give an introduction to neutral reflectometry slide, and I did remove almost all slides from this presentation except this one that I'm not going to use to explain you how a neutral reflectometry experiment works, but it just allows me to show you an example of data set for the supported bilayer with Corée, where you can see some nice fringes in the data because we were able to really load quite a lot of protein in the bilayer, and this protein, as I mentioned before, is also fairly large and rigid. So this was our very good proof of concept for this sample preparation method. But what I actually wanted to highlight with this slide is the fact that the investigation of this membrane proteins with neutral reflectometry is actually quite relevant because not all membrane proteins are rigid. Most of them contain flexible domains. This was the case for tissue factor, for example, where this protein domain could freely move at the membrane surface. And when you have such a situation, interpreting the neutral reflectometry data could be not that straightforward. And it becomes quite important to have other information available. For example, a combination with MD, it's quite valuable because you can picture the dynamic of the membrane protein and these movements with respect to the membrane surface and try to extract from that the corresponding reflectivity curve, then to compare with the data. And together with Andrew, we have been discussing this a bit, and I think it's something that is already out there. There are groups that are doing this, but of course it would be nice for this to become something a little bit more standardized in the future. And then this could actually allow to study quite a lot of different membrane proteins and check, for example, how their orientation can change depending on the lipid composition or other parameters such as pH, temperature, or binding of ligands. But I will not say more about membrane proteins actually, and rather show you something about compatibilities of this sample preparation method with a different lipid system. So these were the very first set of data that we collected when we started working with these peptide discs. And of course, we first started with a very simple model system that is just a POPC phospholipid. This is a very well-studied phospholipid, and there is plenty of data out there for supported lipid bilayer made out of POPC. So it was a good system for us to compare our sample preparation method. And here you can see the data that we also collected then by mixing POPC with other phospholipids, for example, POPS, that is negatively charged, or POPE, that it's zwitterionic as POPC, but with a different chemical structure, and mixture with cholesterol. And altogether, these, I would say, represent pretty standard supported lipid bilayer that are often used as models of a very simple model of biological membranes. In all cases, as you can see from the scattering length density profile, we could prove that supported lipid bilayer was efficiently formed with structural parameters in agreement with what was expected for this chemical composition, and most importantly, with good surface coverage. And I think at least the POPC lipid is now also in the SLD database, and I will try to add also all these others, since they, as I said, they are also very common system used for neutral reflectometry experiments. But as I mentioned, using 1 or 2, 3 phospholipid mixed together does not really provide you a system that resembles too closely a biological membrane. So we also investigated the possibility of using more complex extracts. And this is where using the peptide disc became quite useful, because when you try to form this supported bilayer with natural lipid extract and using conventional methods such as vesicle fusion, it is quite difficult to obtain good qualities bilayers. Most often you have to heat up the sample up to 50 degrees, and this can create some chemical damage to some chemical modifications to the lipids. In our case, I collaborated on this with ILL and the lipid lab that they have there, where they have established now quite reliable protocols for growing Pichia pastoris cells, which is a yeast, both in deuterated and hydrogenous culture media. And then they can extract and purify phospholipids from these cells, which produces phospholipid extracts, both in their hydrogenous and pure-deuterated version. And as you can see from this chart here on the right, these extracts actually contain quite a large variety of different lipid species that differ for different composition of their head groups, but also for different length and unsaturations of the lipid tails. And here you can see the data that we collected both for the hydrogenous and the deuterated extract using the peptide disk as sample preparation method, and once again obtaining satisfactory results for the production of the good quality supported lipid bilayers. So in this last point of my presentation, I would like to show you how we used this natural lipids to form supported lipid bilayer and investigate interaction with the spike proteins, and also finding how actually having this more complex membrane model is valuable in order to study this kind of processes. So as you probably know very well, SARS-CoV-2 is an enveloped virus, which means that the genetic material, the RNA, is encapsulated in this envelope, which exposes at the surface all these little tentacles that are actually the extramembrane domain of glycoprotein known as spike protein. And what was well known is that the spike protein targets a membrane protein that is normally exposed this enzyme has a viral envelope and the cell membrane. And this membrane fusion event is actually what allows the virus to release its genetic material inside the cell. And this is how the viral infection starts. So while this protein-protein interaction was actually well characterized, there were less information about what impact this spike protein could have on the surrounding, on the lipid membrane that would normally surround the ACE2 enzyme. And this is actually what we wanted to study. So to do this, we used two lipid systems. We had a standard synthetic system consisting of POPC and POPS that we used as a reference. And we also had a much more complex membrane containing the lipids extracted from the yeast and also deuterated cholesterol provided by ANSTO as a more realistic model of the plasma membrane, so of the most outer cell membrane. And we use these two kinds of lipid mixtures to form supported lipid bilayer with the method that I explained earlier, so with the peptide disk. And the supported lipid bilayer were prepared in the presence and in the absence of the ACE2 receptor. We did not work with the full-length protein, but just with the part of the protein that protrudes out of the membrane. So in the first part of the experiment, there was, of course, a sample preparation and characterization of the membrane. And then we added the extramembrane domain of the spike protein. So the spike protein that you see represented here is actually a trimer. So it has 3 identical monomers assembled together. And it's actually very difficult to express in its full length version. So what we had available was actually the extramembrane domain. So the part of the protein that actually protrude out of the virus. And it is also where the protein exhibit its domain recognizing the ACE2 receptor. So this is also the part of the protein that would normally closely interact with the host cell. And this protein was produced at the IBS in Grenoble. So here you can see the data that we collected for the synthetic lipid system. So up here is just a POPC, POPS bilayer formed with the peptide disk, just as a control that bilayer was formed with good surface coverage and expected structure. And then we added the spike protein, as I mentioned, in the presence and in the absence of the ACE2 receptor. And this is what we got. So when we first looked at the data, we were a bit unhappy with the result because we initially thought that something went really wrong with the experiment, as some of you sometimes may have experienced. If you work with soft matter and just physisorbed molecule, it can happen that by accident, the bilayer goes away because the substrate is not hydrophilic enough. So we started making all possible hypotheses about why this membrane was not formed as expected and why there was no protein among the lipids. But then we started repeating the experiment and consistently obtaining the same results. And in parallel, we also did some QCMD experiments and concluded that what we were observing is not an accident of a bilayer being destroyed by some wrong experimental condition. It was actually the effect of the spike protein. So this somewhat suggested us that the addition of the spike might have produced a strong association of the protein with the lipids, resulting in the protein taking out of the lipids of the substrate. And because you have, of course, to wash your system to exchange contrast, we would also wash out all the soluble spike with some of the lipids taken out of the supported bilayer. Then we repeated the very same experiments, but this time using the more complex supported lipid bilayer, the one formed using the natural lipids. And we observed overall the same outcome. So as soon as the spike was added and data were collected, we observed a decrease of the surface coverage, quite dramatic actually, and the removal of the lipids from the substrate. What was interesting actually is that this effect resulted to be more pronounced in the case of the natural lipid mixture compared to the synthetic lipids, which somehow suggests to us that there must have been some components in the natural lipid mixture for which the spike protein might have a larger affinity. And here you can see a little bit of a summary of the results that we obtained. So in blue, you have the surface coverage for the synthetic lipid system. And you can see that it systematically decreased when we added the spike protein. And here you have in red the one that we measured for the natural lipid mixture, where you can see that the decrease of the coverage is even larger when you have this kind of lipids. So to try and understand whether this interpretation of the data could be effectively correct and not the impact of the spike, we then looked more closely at the structure of the spike protein and discovered that recently in this structure, it was pointed out that very close to the protein domain that is responsible for the recognition of the ACE2 receptor, there is a lipid pocket. And that this lipid pocket apparently likes quite a lot unsaturated lipid, of which the natural lipid mixture is much richer than the synthetic model system. So this in a way brought us to really believe that our hypothesis that the spike could bind lipids and then be removed during the buffer rinsing step was actually the cause for this loss of surface coverage. Now, clearly in a real system, the spike protein will not be a soluble protein. It will be anchored to the viral envelope. So the take-home message is not that the spike comes, takes the lipids and goes away, but it's more that the spike must induce a strong perturbation of the lipid membrane, and having the rest of the viral envelope close by, this would definitely favor the membrane fusion event that allows the genetic material inside the virus to be released inside the host cell. So as a conclusion, I hope I showed you a different way to produce supported bilayer that allows to produce bilayers with quite a different lipid composition, not including just synthetic lipids, but also natural lipids. And I think that this is an aspect that is quite important to look and design properly when it comes to study protein-lipid interaction, because any lipid bilayer is not necessarily the best representative model of the biological membrane that one has in mind. This method can also allow the incorporation of membrane protein. I showed a few examples of that, although I didn't enter into many details. And I think that membrane proteins are in general quite a complex system to study, where further development on data analysis point of view could really help and improve a better interpretation of the data analysis. And then I showed you a more specific example where the spike proteins from SARS-CoV-2 were used in connection with a lipid membrane, and neutron reflectometry allowed us to sort of make a new hypothesis on the mechanism through which this protein could favor the fusion between the virus envelope and the most outer membrane of mammalian cells. So I hope you enjoyed this talk. I would like to thank all the people I have collaborated with for all these projects, in particular my former group at the University of Copenhagen. That is where I started this entire peptide disk project for producing these complex supported bilayers, and also the team led by Giovanna Fragneto at ILL. I collaborated with them for this project concerning the spike, and I leave you with this picture of the Selene Guide 1 installed here at TSS, one of the components of the STI instrument. This picture is actually 1 year old, so the guide was installed in July of last year. But I still keep showing it because now they have installed all around the guide system the bunker wall, so it's very difficult to have a clear view of the guide system this day. And I would like to thank you all for your attention.